Today we will discuss about this Ezrin-Radixin-Moesin or ERP role in immune cells. In the previous video we have understood about this L-selectin structure where we briefly touch upon this ERP, that in the intercellular region ERP also helps in this L-selectin intercellular signalings. So how this ERP plays a role towards these immune cells, that we will discuss in this. And so figure on the left hand side or on the panel A shows different domains of this ERP which is divided into this FREM domain, coil domain, and F-actin domain. This FREM domain, it is on this N-terminus side, and F-actin binding domain is on the C-terminus side. And since it is immune cells, its main function whenever it sees a foreign particles is to activate foreign particles or foreign motifs. It's to activate themselves and act against it. So because of that, this also contains a ITAM domain, and this ITAM domain is in this FERM domain, FERM domain, where once it gets interacted with its receptors, or then it will trigger these activations of the immune cells. And apart from that, this coil domain contains this Y353, which can interact or which helps in the upregulation of this P13K pathway. P13K pathway is normally known as survival pathway. So it helps in the survivals of the immune cells. And apart from that, in the main role of this F-actin domain is to helps in the interactions with this F-actin and through this ROCK pathway. And so this is the different domains and what is the important role of each domain. Now we will discuss how they gets activated and what are their natural state. That is shown in this panel B. So where in the natural form it is usually in this inactive anti-parallel dimer, that we can see that the N terminus bind to the C terminus of the another ERP, and this is how they form inactive anti-parallel dimer. Once it is phosphorylated, then it is converted into the active form, and once it is in active form, it can interact with L-selectin. That can be seen here, that it helps in this L-selectin intercellular signalings. However, there is a problem that its main activations is mainly on the serine side. So when those kinase is upregulated, that is serine, threonine kinase are upregulated, then it might cause the deactivations of this ERP. And one more way that this ERP is deactivated is been reported or find out that when CXCR4 interact with this CXCL2, then it will also cause inactivations of this ERP. And this is found in the T cells, that this is how the ERP responds when the CXCR4 interact with the CXCL12. And its main job is to help as a linker that connect the plasma membrane interactions to the cytosols intercellular signalings or cytoplasm intercellular signalings. So, and this part, that L-selectin's part, that we have discussed in the previous video, where we have discussed what are the different domains of the L-selectin, and it is a type 1 glycoprotein, that is N-terminus is on the extracellular side and the C-terminus is on the intercellular side. And so basically this ERP interacts with this C-terminus region, thereby helps in the intercellular signalings. And also that we have seen that its FERM domain contains the ITAM sequencing that is shown here, and this ITAM sequencing helps in the activations of the immune cells. And as we have discussed, that L-selectins are also present in the band close form, and which needs to be open up once they are interacted, once the intercellular interactions occurs. So this is how the ERP plays a role, that its main job that people consider is act as a linker between plasma membrane receptors and the intercellular signalings. I hope you understood at least the basic role of this ERP or Ezrin-Radixin-Moesin in immune cells. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.